Connaissance,
Recherche,
sciences,
Des mathématiques,
théorie,
physique,
philosophie,
Conférence,
archéologie,
nature.
So, Mesdames et Messieurs, ladies and gentlemen, vaccines have done more to save lives than any other modality except for clean water. May I have the slides? From Jenner's empirical discovery that cowpox prevented smallpox and Pasteur's discovery of attenuation in the laboratory, there are now numerous vaccines. For example, in the US, there are over 30 vaccines licensed, not counting combinations. The use of these vaccines have had striking effects in public health. Again, using the US as an example, the cases of diseases for which vaccines are available have declined tremendously. This slide shows the peak cases before vaccination, before introduction of vaccination, and cases in 2009, and the profound reductions that have been seen in these diseases. These same effects have been seen in all developed countries. In addition, one of the outstanding public health achievements of the last century was the increase in vaccine coverage in developing countries due to the expanded program of immunization promoted by the WHO. Smallpox is now a memory, and the incidence of polio and measles have been lowered to the point where elimination or eradication is at least a possibility. As you can see, from inception of EPI to the end of the century, we achieved about 80% coverage of infants in developing countries. However, as shown in this slide, there are still many diseases uncontrolled by vaccination. How are we going to develop vaccines against these diseases? Vaccines are commonly divided into attenuated and inactivated vaccines, although as we shall see, the distinction is becoming somewhat blurred. This slide shows the evolution of attenuated vaccines. After the vaccines developed by Jenner and Pasteur, in the 20th century, two important vaccines were developed by passage in animals, eggs, or in vitro: yellow fever and BCG. After the Second World War, Anders Weller and Robbins discovered how to grow viruses in cell culture. This led to the selection of mutants through adaptation in cell culture. These mutants proved to be attenuated, and so we had measles vaccine, oral polio vaccine, and others. It was also found that passage at lower temperature of incubation allowed for faster attenuation and selection of attenuated mutants, and that led to rubella and more recently live influenza vaccine. Now, this slide shows some of the newer strategies for attenuated vaccine development, including reassortment, reverse genetics, recombination, codon deoptimization, etc. I can only illustrate some of these. Reassortment is possible when viruses have segmented genomes, as in the case of rotaviruses, the major cause of infantile gastroenteritis. The virus has 11 segments of double-stranded RNA. So in order to develop one of the vaccines available, we took a bovine rotavirus, which is attenuated for humans, and on the surface of rotaviruses there are two important proteins, VP4 and VP7, both of which carry neutralizing epitopes. By co-infection with four common human serotypes of rotavirus, and then selection of one could call them hybrid viruses, in which 10 segments of the bovine virus RNA were retained, and one segment of coming from the human viruses, that segment being one that coded for either a VP4 or a VP7. Thus, one obtained a pentavalent vaccine giving responses to a broad range of VP4 and VP7 proteins. This vaccine, together with another vaccine, is now being widely applied and will have a major effect on mortality due to diarrhea and dehydration in the developing world. It has already demonstrated a profound reduction of rotavirus diarrhea in the US. One of the most difficult problems in vaccine development for pediatrics has been a vaccine against respiratory syncytial virus of infants. One promising approach involves deletion mutants, an RSV virus that has 4 attenuating deletions in its genome is now in advanced clinical trials. The second approach is through recombination. This slide shows at the top a cartoon of the genome of a parainfluenza type 3 virus. This virus was in fact derived by reverse genetics of a bovine parainfluenza virus 3 and recombination with a human parainfluenza virus 3. But in order to develop a vaccine against RSV, the hemagglutinin and neuraminidase gene was deleted from this genome and replaced by the gene for the RSV fusion protein, thus giving a recombinant virus. This virus was then inoculated into monkeys and together with a control group, and then monkeys were challenged intratracheally with wild RSV. As one can see, the placebo monkeys developed considerable virus in the nasopharynx and in the lungs, whereas the immunized monkeys resisted this infection. Oral polio vaccine strains revert to neurovirulence in the intestine, as Dr. Wimmer undoubtedly will discuss with us. A strategy has been suggested for obtaining more attenuated live polio vaccine viruses using codon deoptimization. As you know, amino acid pairs are coded by more than one triplets of nucleic acids, and for example, the most common for the virus is this set of triplets, least common is this set of triplets, and the strategy is simply to mutate from the most common to the least common in order to attenuate the virus. The result is decreased neurovirulence and decreased reversion. An important current strategy in experimental vaccinology is the use of vectors, defined as non-pathogenic vehicles into which genes from pathogens have been inserted and which express those genes. Many viruses and also bacteria, by the way, have been used as vectors: pox viruses, adenoviruses, VSV, AAV, alpha viruses, CMV, and measles. One use of the vector strategy, which is currently being applied, is to develop a vaccine against dengue. Dengue is a flavivirus with 4 serotypes, widespread in tropical Asia, Africa, Latin America, and the Caribbean. It causes fever, of course, but frequently with fatal hemorrhages, the so-called dengue hemorrhagic fever. The latter may be caused, is thought to be caused, by heterotypic antibody-dependent enhancement. And therefore, one must immunize against all 4 serotypes simultaneously. The strategy used is shown on this slide. The yellow fever 17D virus, used as the vaccine against yellow fever, is the vector. And here's the cartoon of the genome of that virus. The pre-membrane and envelope genes from yellow fever are removed and exchanged for the corresponding genes of each of the 4 dengue viruses, so that one ends up with a chimeric complementary DNA, which is transcribed to RNA, and the RNA used to transfect cells in culture. This, of course, allows one to grow these 4 viruses in culture to develop a tetravalent vaccine, which is now currently in phase 3 in tropical countries. Dr. Griffiths will give us details of the effort to develop a vaccine against cytomegalovirus. This virus is of interest for many reasons. The most important, the number 1 congenital infection, now that rubella has been controlled, although often inapparent, leading to deafness and mental retardation. As Dr. Aspinall will probably tell us, it also may have a role in immunosenescence, certainly of interest to me. But for an effective vaccine, one may need both antibody and CTL, although antibody strategies are currently quite promising. One of the 2 main protein targets of CTL is the pp65 tegument protein of the virus. By insertion of the gene for that protein into a canary pox vector, one could then inoculate seronegative volunteers, and all of whom in this study, shown in the black triangles, developed strong CTL against the virus. So if one will need to develop a CTL response as well as antibody, the vector strategy will clearly be successful. Turning now to inactivated vaccines. The simplest strategy to develop an inactivated vaccine has been to inactivate the whole microbes. And this started in the late 19th century, but continues today, giving us vaccines, for example, against hepatitis A and Japanese encephalitis virus. In the early 20th century, it was the researchers here in Paris as well as elsewhere showed that certain bacteria produce toxins. Later, it was found again in France that one could inactivate the toxins to form toxoids and that these would be powerful vaccines. Also, one began to take apart the organisms, either by extracting proteins or by breaking down bacteria into polysaccharides as well as proteins. The polysaccharide vaccines turned out to be quite powerful against some bacteria, but only in children and adults, but not in infants. In the 20th century, we found that by conjugating a protein to these polysaccharides, one could then induce a T-cell-dependent response in infants and protect them against invasive bacterial infections. These infections then have been virtually eliminated in developing countries that use these vaccines. This slide shows some new strategies for inactivated vaccines. Replication defective particles, DNA plasmids, DNA shuffling, reverse vaccinology, and some of the omics, which I will discuss in a moment briefly. So for replication-defective virus-like particles, as I'm sure most of you know, this is the strategy used to develop the human papillomavirus vaccine, based on the point that a protein called L1 is the predominant protein on the surface of the papillomavirus virion, and that by inserting the gene for L1 into yeast or into baculovirus, and growing the baculovirus in insect cells, one could form virus-like particles shown on the right part of this slide, which turned out to be extremely immunogenic, and the vaccines developed using this strategy have given virtually 100% effectiveness in the prevention of transformation of the cervical tissue or other mucosal tissues by the papillomavirus. Papillomaviruses, particularly the 16 and 18, that are the most oncogenic. Well, these vaccines, however, are expensive, and one interesting strategy which may be useful for developing a vaccine for poor countries is to insert the gene for L1 into the TY21A Salmonella typhi vaccine, which was attenuated by auxotrophy and which is administered orally. So again, this gene inserted into TY21A, and at least when given to mice, the bacteria infected the intestine and produced virus-like particles in the intestinal cells. The mice, given this vaccine, developed serum and vaginal antibodies to the papillomaviruses. Now, the VLP strategy is being used for many other viruses, including influenza. In this case, the genes coding for the hemagglutinin, neuraminidase, and the M1, actually the matrix 1 protein, were put into baculovirus. Then that virus infected insect cell culture, and this is the fluorescence showing the expression of the antigens. And these proteins spontaneously form virus-like particles. So this is a method of producing an influenza vaccine that does not involve eggs and the problems associated with growing viruses in eggs. And this vaccine is actually in phase 3 trial in North America at the moment. Years ago, it was found serendipitously that injection of genes carried in DNA plasmids resulted in translation and transcription into proteins that would elicit an immune response. This form of vaccination has some interesting advantages. It is a very good prime to the immune system, including the neonatal immune system, and at least experimentally, it can overcome suppression of responses by maternal antibodies. It orients the neonatal immune system towards a Th1 type of response. It is very good at inducing immunological memory. However, the antibody responses to DNA vaccines historically have been poor, and a lot of DNA has had to be used. This situation has recently changed. It's changed because one now has at least 2 ways of enhancing responses. One is the use of electroporation, a small electric current given at the same site as the injection. The other, shown in this slide, is the use of a cationic lipid-based adjuvant, which is formulated with, in this case, measles, hemagglutinin, and fusion proteins, DNA, and the DNA vaccine injected into primates gave a response which is clearly protective. Actually, the monkeys were challenged and they were protected. But these levels of antibody are similar to those achieved in humans with attenuated measles vaccine. So this is a major step towards the utility of DNA vaccines, particularly when one needs an antibody response. A strategy that has been now very important, particularly in experimental HIV vaccines, is the prime boost strategy, but this is also being applied in other domains. This is an illustration of what is meant by a prime boost. In this case, the two groups are a pox virus, Nivac, related to vaccinia alone, this Nivac carrying genes for HIV, coding for these proteins, versus a DNA prime followed by the pox virus. So in white you see the responses, or rather the percentage of responders by ELISpot to the Nivac alone, and here is the prime boost DNA followed by Nivac, and it's obvious in the black bars that there is a profound enhancement of the response by using the DNA prime. Vaccine manufacture is also changing. We have new substrates for vaccine antigens, not simply primary cells from animals, but also cell lines spontaneously derived, for example, Maiden-Darby canine kidney, but also cell lines derived by transfection using the adeno5E1 gene. So we have human retina cell lines, chick embryo cell lines, which are proving extremely important in improving yields of viruses in production of various vaccines. Insect cell lines, as I've alluded to, using baculovirus vector, and also plant cells using an agrobacterium vector. And I illustrate this. The plants are grown in the laboratory, and in this case we have what is actually a beneficial use of tobacco. These are nicotine, that is tobacco plants, grown in mass in a sterile laboratory. And one then puts the gene of interest into an agrobacterium and infiltrates into the leaves of the tobacco plants, the agrobacterium vector. And in this example, the vector was carrying the gene for green fluorescent protein, and you can see the profound fluorescence of these leaves, which are then harvested, ground up, and the protein purified, and this has been applied to vaccine proteins so that one can produce them in quantity, which obviously has important economic implications. Another major advance in vaccinology is the use of adjuvants. And here is a short list of some of the adjuvants used in vaccinology. The old ones, well, relatively old, oil and water, so-called MF59, water and oil adjuvants, but also now we have saponins, we have liposomes, bacterial toxins, particularly for mucosally administered vaccines, cytokines, and perhaps the most important these days, TLR ligands. Ligands that induce responses of one or the other of the TLRs. In particular, this type of vaccine, TLR4 agonist, has recently been licensed in the US as well as it has been licensed in Europe. And there are others which very likely will be licensed. Another aspect of adjuvantation is the use of nanoparticles and nanoemulsions. These may be particularly useful for vaccine delivery on mucosal surfaces, for example, intranasal vaccines, and also for incorporation of adjuvants together with antigens in particles that are highly immunogenic. And the route of administration is also going to change. The situation today is we are giving many vaccines by parenteral, that is intramuscular or subcutaneous injection. There's a limitation to the number of injections one can give, even with combination vaccines being developed. The transcutaneous route is possible because if one, for example, puts in a patch an antigen together with an adjuvant that stimulates diffusion through the epidermis, the antigen can reach the Langerhans cells shown in red here, and of course those cells go to draining lymph nodes and elicit an immune response. So this type of strategy has been applied, for example, for vaccination of travelers against travelers' diarrhea caused by E. coli and Shigella, etc. In addition, there is the use of microneedles. Now, microneedles are relatively painless, and so one can administer quite a few vaccines this way. This is, you can see the small needle at the end here. And before I go to that, just let me say that this has been applied now to influenza vaccine, and I believe this route is now licensed in Europe and is likely to be widely adopted elsewhere. Now, then, turning to the omics, the first and most important thus far is the use of genomics, or otherwise called reverse vaccinology. This consists of obtaining the whole genomic sequence of a pathogen, analyzing the sequence for DNA open reading frames that are likely to code for surface or secreted antigens, expressing those ORFs in E. coli, immunizing mice with the proteins expressed in E. coli, using the antisera from the mice to confirm the localization and the bactericidal activity. And if that antigen is conserved in different strains, one would begin clinical development of a vaccine derived from those proteins. The most advanced application of that strategy of reverse vaccinology has been with group B meningococcus. This has been a problem for vaccine development because the capsular polysaccharide of group B has homology with neural polysaccharides in humans, and therefore one is loath to use the capsular polysaccharide as the vaccine. But a vaccine composed of 5 newly discovered proteins from the meningococcus is now in phase 2 of development. But in addition, many other bacteria, group B strep, pneumococcus, anthrax bacillus, chlamydia, and Porphyromonas gingivalis, the cause of periodontitis, are all the subject of reverse vaccinology, and this strategy is yielding vaccine candidates that look promising. There are other omics, transcriptomics, for example, which is the search for messenger RNA expression in vivo, because if a protein is produced in vivo, it is likely to have some pathogenic significance, and so possibly the subject of a vaccine development. In addition, there is proteomics, which is the characterization of microbial protein function and interaction with nucleic acid, lipids, and other proteins. And lastly, antigenomics, which is actually burgeoning at the moment. That is the identification of antigens to which immune responses are induced in convalescent sera, again, likely suggesting that those proteins are important. Lastly, microarray, or so-called systems biology, is now coming into use. And I give you one example, actually the first example that I'm aware of, in which it has been possible to predict, or at least retrospectively predict, what vaccines are likely to work. In this case, it was a malaria vaccine, and it was possible to challenge the vaccinees with malaria and to look back and see what responses they had after vaccination that correlated with protection. And it turned out in this case that 4 genes were expressed in those who were protected, that is, white triangles in this case, and not in the asterisks, which were the unprotected individuals. These were genes associated with HLA peptide processing. So very interesting and logical, perhaps, so that it is now possible, at least with malaria, to look and see what genes are upregulated by a vaccine and then to try to increase that upregulation to develop a better vaccine. So there are many improvements in vaccines for major targets. For influenza, conserved antigens are being added to hemagglutinin. For HIV, as I think we'll hear, we now have the observation of 30% protection after a vector prime protein boost, and certainly we need better vectors. For malaria, we have 50% protection in infants with circumsporozoite protein in hepatitis B virus-like particles, and it's apparent we need additional parasite antigens or better adjuvants. And for tuberculosis, to prolong protection by BCG, immunization with Mycobacterium tuberculosis antigens carried by vectors, for example, including BCG itself as a vector, this is hoped to be able to vaccinate adults. Well, I won't, in view of the time, I won't tarry on this slide, but only to say that there are still many important problems facing us in the 21st century, problems that are vexing, such as the post-maturity of the immune system and our need to vaccinate the elderly as well as the young. But these problems are problems that I think are soluble. If we go back to Émile Roux, who said, "La science nous apparaît calme et triomphante quand elle est faite, mais la science en formation n'est que contradiction et tourment, espoir et déception." But, et pourtant, I think that vaccinology has been a successful mix of microbiology, immunology, epidemiology, public health, and now molecular biology. And it has become a science in its own right, and in this century is going to yield many new and important vaccines. Thank you.